I think yesterday went really well on all counts. A couple of programmatic notes. We're going to start off as scheduled with Alejandro, followed by Jay, and then Matt, and then we'll finish with Preeti's presentation. So I haven't left anybody out, right? And Vinay Pai will be our moderator this morning, and I think that should cover it. If you have any travel questions, Donna, is at the moment not sitting there, but oh, there she is. Like Donna stepped out. So for travel questions, Donna sitting over there is your connection. And with that, I think let's get started.
All right. Before I get to the mammography part of my talk, I'll describe another visual search, namely that for camouflage objects that led to the mammography work, and I'll talk about the mammography work during the third hour of my talk. About camouflage is searching for camouflage is searching for objects that are hidden in plain view. Kathleen showed examples of that yesterday. And the way we do it in the lab is we synthesize camouflage images so we know what we put in. We start with natural textures, in this case mushrooms or foliage, and we use Portilla and Simoncelli's steerable pyramids to synthesize additional examples of that background. And we take one of the backgrounds and put, in this case, a face target. It could be any of these, and in this case, it is this one. And so this is the scene that the subjects look at. They're given them one at a time, and they have to, in this case, perform a very simple detection task. And the reason for synthesizing the image is that we know the ground truth because we either put in the target or we don't. We can precisely manipulate the scene features because the steerable pyramids are essentially a multi-level image transform. And make the task truly statistical in the sense that the subjects never see the same image twice, so they cannot do the task by remembering any of the pixels or episodic details of the image. They have to learn what's common about the background. And the data I'm going to show you are from one of the simplest experiments published. There's a single target, no distractor, or clutter, and the task is, as I said, a very simple detection task. Is there a target present or not? The intuition is that if you know the statistics of a given scene, if there's something in there that doesn't belong, you should be able to use an odd man out strategy to report if there's something there that doesn't belong in the detection task. So here are the results. So before training, the subjects perform at the chance level, and this is their performance over various training blocks. And in the post-training, so their performance continues to improve. This is statistically significant. This is one of the earlier set of results, but now we can routinely get D prime 2 or higher. Once the subject learn a particular background, if you give them a novel target, in this case, we call these digital embryos. These are novel objects that we can be certain that the subject has never seen before, and they have no problem finding it because they know what the scene ought to look like, and they can tell the odd man out, so to speak. But if you use another background that is that subjects are not familiar with, and it is unrelated to the original background, there's no transfer, there's no improvement in performance. And this is average across the subject. Now, I said it doesn't transfer to unrelated backgrounds, but we have done additional experiments where we have taken an image like this. So if this is learned, obviously it transfers well to additional instances of the same background. But if we simply systematically manipulate the texture statistic like this, so this is in the texture space, it's orthogonal, and the transfer is poor, and over the tuning data points I showed you, the performance transfer monotonically decreases. And so one of the intriguing findings, recent findings we've had, is that the probability of microsaccades significantly increases during the pre-asymptotic phase and significantly decreases during the asymptotic phase. And here's what I mean. So microsaccades are these small eye movements, non-volitional eye movements that are under a degree or less. And so this is the one subject eye movements when the subject was not trained. This is the asymptotic phase. Notice both the saccades, a lot of things are happening here, but we are interested in microsaccades precisely because they're non-volitional. The number of saccades also go down. And it was alluded to yesterday, when the subjects become experts, they may tend to make very few saccades, and also the frequency of occurrences of microsaccades go down. And this is summarized here for one subject. The red dots are the D prime, the subject's past performance, and the blue diamonds are the frequency of microsaccadic epochs. It goes in, goes up, and then shows this characteristic V-shaped curve. Finally, oops, I think as Brandon was alluding to yesterday, search CAS makes a huge difference, and we also have a whole lot of fMRI and concurrent fMRI, EEG, et cetera. We know that other parameters such as crowding, et cetera, also make a huge difference. Now, about the mammogram work, we just wanted to see whether we can use, and to what extent whether you can use such an approach. And we use the same, we use several methods. You can also do, a transform like principal components analysis. In this case, this is the original image, and this is the synthesized image using the steerable pyramids method. And these are the data for one subject. And so the point is, using this method of mammographic data, we can train them. These are people off the street, both in camouflage and mammography. They become pretty good at looking at detecting an object that doesn't belong. And here's just another method, the image analysis method of Aaron Hertzman of NYU. This is the original mammogram, screen film mammogram, and these are a couple of synthesized examples. We are able to change the viewpoint from CC cranial caudal or MLO. And so this is one of the things that we're interested in. Change the various parameters, viewing parameters that the radiologists are to contend with. I'm done.
So I didn't catch, when you do transformations and tests for generality, can you change it in a way that maintains the overall scene statistics, like scramble it, and does that transfer well?
Can you scramble the image?
Yes. So what I'm after is whether what they're learning are almost like, learning specific feature. Like if it's the exact same background over and over again, you can learn.
Machine learning sense, we know those are the features. We have not tried scrambling because, as you know, scrambling introduces information, usually noisy information, but it also, essentially changes the transform. We know that because what we put in are basically values along the basic functions. We know those are orthogonal by definition. And we have done experiments. I didn't show you the data. If you add noise along an orthogonal axis, if it is actually used as information, it should affect performance, and it doesn't. It acts as noise. And so what we expect theoretically does pan out psychophysically.
So do you, you were doing this in free viewing, right?
Right.
Do you have any idea whether or not your trained subjects can beat chance in a single glance without an eye movement?
Yes. In fact, they can, after training, after they become experts, they trained subjects don't even make eye movement, even when given a chance.
And do you, are trained subjects localizing the target, or are they simply picking up on the disruption of the statistics?
We have done both, and even if you have them localize the target, and even if the stimulus presentation, this is for camouflage study now, if the stimulus duration is as short as 50 milliseconds, they can still do the task at deep final, highly significant overlap.
Okay, but so when they're doing it with the brief exposure, are they localizing, or are they doing this?
In this particular example I showed you, they are simply reporting whether there's a target or not.
Okay.
But yeah, we have done experiments where they are actually localizing.
Yeah.
That data for which I did not show. Yep.
So I have a quick question. You said that they learn the background. When you use a novel target, learning transferred, but they actually started out at that same low deep prime. If you expected transfer, you'd expect it to kind of pick up where learning asymptoted.
Are you referring to the bar plot that I showed you?
Beginning.
Right. That was an example where we were testing a texture that they were not exposed to begin with, and it was on an orthogonal axis. So in other words, it was truly an unrelated background texture.
No, but this is the first plot had, I think it was the blue symbol. Yeah, blue line. So the blue line starts again there. I mean, if there was real transfer, you should expect.
No. So this was basically, we tested them before they were trained for both the novel target and the phase target, and then we did them, trained them in the background, and then we tested them in novel and
Okay, if camouflage breaking was due to just a local disruption in the background statistics, how would that relate to the satisfaction of search stuff that we talked about yesterday? It would seem that the second target would pop out equally well.
And in fact, I didn't show you the data, and in fact, that might be happening. Yep. In fact, in other words, if you, the crowding effects are roughly along the lines of satisfaction of search.
Okay, that'd be interesting to see that. I'm very curious.
Yeah, but you would kill me if I showed additional data.
Okay. But also with respect to your microsaccade data, those are really cool. And I was wondering if you're thinking about this in terms of some sort of way of evaluating when maybe radiologists have been sufficiently, trained whenever they stop exhibiting these microsaccades, because my inference is that these microsaccades are indications of them still learning the background statistics.
Right. In fact, we're thinking of doing that. My radiology colleagues, Dr. Norm Thompson and James Rawson, have been very cooperative. We indeed plan to do that.
Very cool.
I just want to add one other thought. The claim here is not statistical learning explains everything, but it explains quite a bit. So there's a very good question as to how much, whether it explains with all the relevant non-random information or not. We do not know the answer to that.
So, I wonder if maybe I missed something, but with respect to your microsaccades, I wonder if you think of them as having a functional, playing a functional role in establishing the segregation signal, right? It's like if I move my eyes a little bit, I'm going to have more variations. It allows me, the visual system, to pick up more of the variation of this uncertain world. And once that it's learned, it falls up. So that means that it would be very much like a thin organization parameter.
Right. Yeah. I agree.
So I'm puzzled by the mammogram learning results.
Okay.
Am I interpreting that correctly? You're saying that the Portia-Simoncelli texture synthesis of mammograms captures all the lots of the statistics of actual mammograms?
No. I hope I didn't say that.
Because I wasn't sure how, like, what.
No.
First of all, transferring real mammograms.
No. Once you see enough of them, you develop enormous respect for the complexity that radiologists have to contend with. No. This is a particular class of screening mammograms. Screening mammograms are mammograms in which the patient doesn't have any previous known breast health complaints, and these are so-called magnification views thereof. Okay, and so it doesn't have the breast outline and the various things that come with the 3D curvature of it. So this is essentially a flat 2D picture, and the algorithm does very well, tweaked by us, because remember it's a shearable pyramid, and the algorithm, and so we had to tweak it to basically get at the right bandwidth. Once we do that, it does a pretty good job. And in this case, the input image data set are fairly homogeneous, picked out for that reason, and the malignancy in this case was an undefined mass.
One quick question is that, or a quick comment, I guess, is that we're, I think, thinking a little maybe too simplistically of expert radiologists go right to the target, and it's a very simple thing. I know, for example,
I didn't say that about
you didn't, but
yeah.
I hear that in the discussion. I'm not,
Yeah.
but I just want to say that people who have studied radiologists and that oftentimes find other behaviors, and in particular, I'm thinking of Claudia Mellencamp, who finds that even expert radiologists will oftentimes go to a lesion and make a series of what she calls stellate saccades, which is basically, is that thing really different from the background? And so they're trying to discriminate it from the local background, so the saccades are being driven by, or at least that's the hypothesis.
Right. No, I understand that. And that is also, Jeremy talked about drillers and the scanners and so forth. So there are the different kinds of search behavior. We see that in camouflage. Hopefully that some of that came across using from our eye movement data. There's a whole lot of going on, whole lot of things going on, and what we present is necessarily, if we have to present it in a little figure, is simplistic for that reason. But there's a whole lot more going on.
Thanks.
I thought you were going to talk about Art's results, how this goes with that sort of, and then the channel isotoning and background statistics. No, that's not worth mentioning. Is it? Well, this goes well with the body of literature. That studies. We've come to, there's just a lot of studies that were after, not exactly this, but a similar ideas, which I mentioned in my talk, which is whether people and radiologists take into account the statistics of the background. And I think there's been a lot of psychophysics, including classification images and stuff like that Craig has done. And the idea that indeed this is one of the things that we haven't assessed well the learning of it, but indeed when you become good at this, you are actually taking into account your templates, how you weight different frequencies, takes into account the statistics, not only just the spatial frequency. There are also studies by others that actually if the noise is oriented a little bit, so it's not isotropic in orientation, humans or people that are good at this are actually also differentially weighting different orientations, sort of orientations that are more noise-free. So not only just the spatial frequency, but all of that comes into account, and they learn that and they adjust their strategy to sort of use parts of the, where there's signal information but there's low noise. So this ties really well with a lot of that. So.
Thanks, Jason. So Todd, did you want all the talks to go back to back, or do you want to have a discussion time? A few minutes discussion. Okay.
at a first degree of approximation, I think that one thing that you can do is you can imagine that when you're trying to accumulate evidence in favor of, right, the template that you're looking for, you could do it. In my experiment is easy because you have very good segmentation of the object at a particular location. But I've talked to other people who do just scene analysis, not based on objects, because as you know, they do just overall features and convolutions and things like that. And you could basically run something very similar, because it would be just a little bit more noisy. But you can imagine doing the same sort of process, and instead of doing it over an object, you would do it over a patch of the image, and you could do it at different spatial scales. And I think that one of the things that is interesting is that as you develop expertise with a background, what you might be able to do is learn the characteristic of this background. So that allows you to reject that as background.
Yeah, it would be interesting to take those texture synthesis things and actually have perhaps two different sets of textures in the same background, where where you learn that the target lives on this kind of background and not on that kind of background, and take a look at the eye movements. And you would probably pick this up implicitly, find out if the eye movements start restricting you to the set of statistics that goes with the background where the target lives. You've already done it.
Yes, and then and the eye movements. I think Jeremy has a terrific intuition, and he's exactly right. And generally, if you create a saliency map, not a contrast map, but some kind of saliency map, the eye movements are the high saliency regions essentially act like attractors for eye movement.
I'm a little confused by the suggestion. Wouldn't that piece of background then just become a primary target followed by the secondary target where the little guy lives?
Well, it sort of depends how you think about what you might call, scene guidance. If I'm looking for water pictures, in some sense, I suppose you could say yes, horizontal surfaces and tables in this room become a sort of a primary target, and the pictures become a secondary target. Or you could think that the structure of the scene is constraining where you're going to search for actual targets. We've done this with where we constrain different types of targets to live on different, basically, colors of background. Found that the eye movements constrained themselves to the right neighborhood, and then when we asked people after the experiment, gee, where do the targets live? And they all lived on like red backgrounds or something. They had absolutely no idea, but their eyes sure had an idea.
Contextual queuing stuff is you get these overall average response times, but if you actually look at the eye movement, it looks like it's really just a small, granted this is a different task, a small proportion of trials that's actually corresponding. It's either, an extra 10% of the time people recognize and they have super fast reaction times, or else it's like they don't recognize the scene at all.
And the contextual queuing one is with repeated, and with repeated displays of the same set of T's and L's, whereas the texture one is different texture. You're learning something about the scene or the texture, but you're not learning about the specific configuration, right? You were changing texture. You were changing texture on every scene. Right?
So
one other thing, we also did an experiment to see if it is just properties of the background, because that's the thing that we ended up studying first. But no, it's actually if you systematically change the properties to the foreground, you can show that the transfer is also a function of the overall statistics of the image, including the target object. So it's not just purely background learning. They do know something about the statistics, learn something about the statistics of the foreground as well, which is useful for us going forward in mammography.
So apart from Craig's talk, I haven't heard much about using the kinds of things that you're doing to actually improve image processing to increase the saliency based on psychophysical properties, and so what kind of smoothing or sharpening of an image will actually improve search statistics and presumably work on real stuff. So I'm not in this field, but I would think that this would feed well into that.
So Andrew Maidman used the kind of models to develop an entire imaging system. So.
Yeah, so we actually call this observer-based design, and we're sort of trying to perfect that process. And Barcode, has sort of led that. So we have an industry academic partnership grant, and they're our industry partner. I think I mentioned this a bit yesterday. The cost of creating these displays is enormous. To go to the point where you can sort of go to a foundry in the Orient and have them fabricate a glass panel that is, structured so that it'll act as an LCD display is you're talking about tens of millions of dollars at that point. And then you have to sort of make them, fabricate them, put them into clinics, get people to observe them, start looking at getting feedback. A lot of that is very soft stuff. I feel fatigued or things like this. It's not that, oh, I'm suddenly missing cancers. It's indirect measures of performance. They had a graphic example a few years ago where they increased they would decrease the focal spot size. They were able to decrease the focal spot size in the CRT, but they didn't match the bandwidth of the amplifiers in the horizontal direction appropriately. And so what you end up having was like little black lines between each stripe on the CRT. And in fact, they actually found that although in theory it was a sharper monitor, didn't have particularly worse noise performance, observer performance was worse. And they ended up pulling that product after 2 years. So that's when they actually went to this pipeline. And things that have been done. One of the things we've done is, or that they've done is in an LCD panel like that, they'll tell you the statistics, typically most monitor manufacturers, of what will it take to drive from zero to black, from black to white, or white to black, and it can be 3 milliseconds or something like that. But what they won't tell you is what it takes to drive from a gray level of 120 to a gray level of 122, and that can actually be something like 500 milliseconds. And so what they ended up doing was looking at the impact of this through virtual clinical trials. Basically, where it was identified what were the sort of necessary timings, and then they worked out drive curves to get the LCDs to drive from one level to another at every gray level. And so their monitors are now calibrated at each drive level. So there's big tables, huge lookup tables of, oh, I need to get at this pixel from 128 to 134.
Quick intuition about that you can give us about why that's true?
On the LCD monitors?
Yeah.
It has to do with the driving voltages. It's a nonlinear function, the driving voltages. And so the change in voltage is very small, and so the resultant effect is also, is very slow.
Oh dear.
Yeah. So I mean, so they actually got a monitor approved. They then went on to show that if you take a regular mammo monitor and you view tomosynthesis images on them, they're blurred because you're scrolling through relatively rapidly, and mammo monitors were designed as static images. And you can actually get if you go to the Barcode booth, who's ever going to RSNA and go to the Barcode booth, they have a situation where they'll show you a roving magnifying glass. And if you move the roving magnifying glass, the electronic magnifying glass, over a calcification cluster, it will disappear. Right? As soon as you stop it, it'll be sharp. Right? It'll be a beautiful image of a calcification cluster. You move it, and it goes away. It just disappears because it's the monitor. And so then they'll take you to their new monitor, and they'll show that the calcification cluster persists. That's the price you're talking about? Yeah, that's about right. a pair of those monitors are about $25,000, but a pair of the blurring monitors is about $23,000. So you're getting the sharpness for $2,000. I mean, it's actually probably a pretty good deal. The new monitor that they just came out with, that they did is they have backlight, and the backlight shines on the wall, and depending on the darkness of the screen, the lighting on the wall changes, and so it changes your accommodation so that you get better visual acuity at different gray levels on the display. So depending on how bright the display is, they play your visual acuity to compensate. Another thing they have is they have a ability you can just touch it, and it will highlight that area. You get higher luminance very briefly in that area so that you can get better perception. These are things that are coming out of the clinical trials we're doing.
I'm sort of wondering what's going on with our sort of standard psychophysics with the Gabor patches that are ramping on and off over 500 milliseconds.
I have a stack of CRTs. I have a stack of CRTs.
So CRTs do not have that problem. They have different problems, so yeah. Well, I still have 2 CRTs in my lab, so, 5 megapixel.
Okay, so I've been looking at searching for, searching for an unknown number of targets. So, so the question here is, some of you have seen this image before. If you have, you can't participate. The question is, find all the beer bottles in the fridge, and you have 1 second. Okay, how many? Not one, all. You agree with him? I said he was correct. But basically what most people do is maybe you'll look at the beer bottles in the door because they're very clearly visible. Maybe you'll look at the beer bottles at the bottom. But very few people look at these uncertain locations. You're a good beer finder. So this is what I've been looking at. If you give people multiple targets and you don't give them all the time in the world, where do their eye movements go? I mean, some of these beer bottles are so visible you don't need to go and confirm that there is indeed beer there. If the task was indeed to find all the beer bottles or all the potential masses in the image in front of you, and you didn't have all the time in the world. Of course, radiologists do have all the time in the world, maybe. This is the question we're asking, and what do people do? Of course, we don't work with beer in the lab, so this is kind of what we did. Boring stimulus, 5 dots vertically arranged, 6 potential locations. Those are the ones in the circles, and a target could appear in each of those locations independently. So you could have a particular image could have 0, 1, 2, 3, 4, 5, 6 targets. You didn't know. You had to actually localize the locations that had targets. And you had to find all the targets. But you were only given 1 second to do it. So where do people look? That's what I'm asking. And in this particular case, if you're looking for that a string of vertical dots, perhaps it's easier for you to see it in that bottommost image. You could see that vertical thing. The circle at the 10 o'clock position, it's less easy to see. So if you had 1 second, it would probably make less sense to kind of look at the ones you can already see. Instead, it would make more sense to look for the ones that you're not sure about and go and verify if there is or there is not a target there. So we call locations like the one at the bottom highly probable target locations. We call them MAP for short because they're maximum a posteriori locations. The ones in the 10 o'clock position, for example, are uncertain or high entropy locations. They're likely to have a target or not, equally likely to have a target or not. Okay, so this is actually what the display really looked like. The previous one I showed you was a cartoon. There are 6 locations here, and there are some targets. And the beer finder maybe can find this, maybe cannot. How many targets? No idea. I highlighted the targets for you. Okay, so this is a hard test. And what did observers do? They made inefficient saccades. They fixated the locations that looked most target-like. They did not go to the uncertain locations. And we trained them with complete feedback. This is more complete than is shown here. We actually showed them their scan path. We showed them the locations they selected, and we showed them where the targets were after each trial. And that did not improve performance. And I was one of the subjects, one of 8, but nobody improved performance. So we said, okay, specifically, I don't want to go into the model too much, but here's the intuition. What is happening here? Everybody's behaving as if these targets were very rare. So they're always going to these most probable locations. So in green is, let's do blue. Blue is the prediction of a model that goes to locations where the targets are most likely. So I'm plotting on the y-axis the proportion of times the model got it correct, identified all the targets in a trial, versus the number of saccades. So obviously, if you look at more locations, you're more likely to get this correct. And what this graph is plotting is data for a very low prior probability of target at a location, 0.17, much higher than in cancer, but still quite low. So here in green is the prediction of a model that basically goes to locations that maximize information. So I'm going to the most uncertain locations so that you can see in this particular case, the green's slightly better, but not much better. So when target probability or prior probability is low, these two strategies are not very different. Let's go and look at the other end of the spectrum. Here, targets are very frequent. You have 0.83 probability of a target at a location. In the red one is just making saccades at random. That's a random model. You can see that now going to uncertain locations is highly informative, and going to the most likely target locations is even worse than random. And what do observers do in this particular study? They went, regardless of the prior probability, they always went to the most probable target locations. Well, this part's cut off. Observers follow the map strategy. Okay, so why? Maybe the task was too difficult. Maybe observers are unaware of their visibility function, how it falls off with distance from fixation. Maybe multiple target search tasks are very infrequent. But we were interested in this last issue. Maybe the feedback at the end of the trial was not effective. So what we did is we came up with a really simple display. No noise, just targets, very easily visible, no visibility function involved. The targets were blobs drawn from a distribution brighter than the background. The distractors, the ones that had no noise, if you want, were drawn from a distribution darker than the background. Importantly, these two distributions overlapped considerably. So something that was a patch that was close to the background was equally likely to be target or distractor. And these are the most uncertain locations. The question is, can observers go here, and how do we make this useful for their eye movements to go there? So what we did is this. They started there. They saw this display. These are the most uncertain locations here. And what we did is, when their eyes went there, we artificially, as soon as the saccade landed there, we changed the mean value of that disk to the value of that disk to the mean value of the distribution from which it came. So we gave them this artificial feedback. Here we were just checking to see if you got this feedback, this immediate saccade-based feedback, would it make you more efficient? Would it make your saccades more efficient? Okay, and the answer is yes, it does. So here I'm plotting the proportion of saccades that went to uncertain versus those that went to the most probable locations. Here is efficient, here is inefficient. You can see as the prior increases, people are doing much better. And this is in contrast to previously, where regardless of the prior, they kept on with the most probable location. And that's all I wanted to say.
Do you have the performance for the same subject as they changed their strategy? How did their overall performance change, and does that track track with the model prediction? Okay.
So you could do it. I don't have a graphic here. I could pull it up for you. It'll take a little while. So in the experiment with feedback, if you do a trial-by-trial analysis of the proportion of trials, the model's performance, if it were making uncertain saccades to informative locations, and observer's performance, and you see a really good correlation. And if you compare observer performance to a model that's going to the most probable locations, they all do better than the model. Yeah. Uh-huh. So in this, okay. In this particular case, it's artificial, right? They're not going to know anything about the, so not here. But to answer that question, what we have to do is, it seems to take quite a long time for people to get trained. We've now, I mean, so when I did those noise experiments, I also did Gabor's and white noise, and people were equally bad at that. But we've slowly got, so the people who have been with us the longest have slowly gotten trained to make these more information-seeking saccades, but it seems to take a very, very long time.
So I rather, when you put up the list of explanations, I kind of like the one where you said observers don't know their own likelihood function.
Yeah.
And I'm wondering whether your experiment is still, the second experiment is still consistent with that. Because when you show people, so they in this case, not knowing the likelihood function amounts to not knowing the distribution around the mean of any given group in the luminance patch experiment, right? And so if you give them immediate feedback about the mean for the group when they saccade there, you're also helping them learn the appropriate likelihood function for the patches.
True.
So do you know
whether that would help?
Do you think so? To what extent do you think the delayed versus immediate feedback is really just a mechanism for better learning of the likelihood function, or do you think that there's something else there?
That's a very good question, but all of this reinforcement learning, they don't delay the feedback, and I don't think they're giving people any information on, the mean of the likelihood function, and that could be a component. It's a very interesting thing to explore, but I'm not sure that is the main thing. I mean, they were getting plenty of feedback before. It was just completely ineffective. I mean, they knew where their eyes went. They knew the locations they picked. They saw their scan paths, but it didn't transfer into, oh, that's the kind of thing I'm supposed to be looking for.
I have a multitude of questions. I'm going to try to be organized here. But one of the issues here is what people are trying to optimize, whether they're trying to optimize some perceptual experience,
yeah,
or whether they're trying to optimize a payout in the task, for example. So if you tell me that I'm going to get $10 for every one, or a beer, for every one of the targets that I correctly located. At the end of the trial, I need to click on all of them, right? Maybe I will go to the uncertain locations if I know if there's some obvious ones out there. But if what I'm trying to do is learn something about my perception or something, I'm doing this differently. I want to go and, oh, I think my target template is this. I go to a location, yes, that's what my target template is. And so they're not satisfying a maximum likelihood of anything. They're just trying to, yeah.
Yeah, they're not optimizing. I mean, you know, they're not. You're saying, did we look to look at a manipulation? So I gave them. We made a calculation about how much more. We didn't give them money, but we said we would weight. Give more points to saccades that went to uncertain locations, and then after each trial, they saw their score and they saw their score increment. And we told them that the top 2 out of 8 would get, excluding me, would get a bonus. They make a difference. It was just something, especially for the first experiment, it was just too hard. It was not. So maybe I didn't do the experiment correctly, but we did that.
Yeah, maybe you try rewarding them not with a beer for each one of the targets they find, but you get a beer if you find the last target. You have to actually get the number right.
And there,
we actually did that with mammographers. We ran an experiment at a meeting with basically at the exhibit during happy hour, and my postdocs who actually ran this do report that data quality goes down with consumption of alcohol.
At the risk of wasting my last few credits for talking too much, but it's the end of the meeting. Briefly, 2 things. One, a very detailed question, sort of, did you compare a model that does map, but not maximum posteriori, but minimum a posteriori? So you actually go to the location that's least likely, and how doesn't that work quite well when the probability is high?
So, for example, when the probability is high, you could say, I'm just going to look for those. And yes, we did look at those. And that would be also an efficient model to use, but they don't do that either. So we looked at, you know, saccades to non-targets, you know, that they go to the most visible non-target. And that's not what they do either.
And then the second question is a little bit larger, more relevant, more a philosophical question, which I think you share sort of the observation. We've talked about this, which is there is, in the eye movement literature, there are a lot of tasks, and this relates a little bit to medical imaging. There are tasks where humans seem to be quite optimal in how they deploy their eye movements when you compare it to sort of an optimal strategy. And there are tasks, some of these lab tasks tend to show a lot of differences. People have a hard time learning this. But when we look like, you know, things that we've worked in the recent years like faces, I can bring anybody from a subject 1 pool, and they're looking at the optimal place to look at to acquire information about a face. And I can show you that if I force them to fixate somewhere else, they do worse. When I bring them to do these tasks like Preeti, well, if it's the subject 1 pool, they're pretty inefficient. They're all inefficient. So it's very, you know, they have to learn these things, and it's much harder. So it's interesting about, you know, really having to do with practice, things that were involved every day, and when we bring them to these new sort of tasks, it's not straightforward that they'll actually start doing some optimal eye movement strategy.
So is the upshot of that you have to hide a face in every mass?
No, I don't think, maybe, I don't know. I thought I don't know how you solve it, but I think it's something to keep in mind.
I mean, when you're training a radiologist.
Yeah, so when you're training right now, I think it's unclear. Well, we don't know how radiologists are a way to evaluate the optimality of their eye movements. It's an open question. So that's a harder problem, but I think that would be interesting to evaluate how good, how optimize their strategy is. So that perhaps is really, because we don't fully understand that, but that might be the difference between the novice and the expert that we put up yesterday, although we do not fully understand how way these are optimal because the images are complex.
That would bring you closer to more optimal eye movement?
Well, I think learning background statistics, good templates, whatever your eye movement strategy, that will, whatever you're doing, if you're using an optimal one or a suboptimal, that will bring you up in everything because you're able to discard. It's a good question. I don't know. I mean, these are fascinating research questions.
Thanks, Preeti.
Welcome.
So I think now the floor is open for general questions. The last two questions for the
So what would be wrong with taking mammograms, splitting them up into tiles, and showing them at every 200 milliseconds?
Splitting the individual mammogram into pieces, or showing the whole? Well, if you cut the mammogram into little squares, you're losing any useful. You're losing a lot of useful structural information, and that won't make people happy.
Appropriate size squares, or whole mammograms every 200 milliseconds.
Well, so to whole mammograms every 200 milliseconds, expert mammographers have a D prime of about 1 in our hands. What would a D prime of 1 translate to in area under the curve, roughly? Yeah, so, they comfortably beat chance, but they're nowhere near as good as they are at if you actually let them loose on them.
Studied as a function of resolution.
What?
Has that been studied as a function of resolution? I'm just wondering if that's been studied as a function of resolution, like the Zumwal's idea, start off with a low resolution, and then what is the RSVP detection rate at low resolution?
Oh, this isn't RSVP. This is single flash and unmasked. So what we were doing was single flash, unmasked, give us a rating between 0 and 100 on a callback to don't callback scale, and you can beat chance in a quarter of a second. And interestingly, you can't localize the problem at all. Your localization performance is 0. You're reading something global. But and we've never tried hooking them up to EEG and see whether their little brains are. I mean, I think the evidence from the image triage stuff is it works quite nicely if you're looking for. I mean, the classic example is heliports, right, with a great big H on them, and it doesn't tend to work that nicely once you start getting more complex. But that may just be that people haven't built the right EEG classifier yet.
Alcandel did both of these with chest X-rays and with mammograms. It's kind of similar results, but they're pretty obvious cancers.
We were deliberately using subtle ones. We were deliberately not using obvious ones. So no calcifications, only masses and architectural distortion, and they were still beating chance, and they couldn't localize it. So it wasn't just that, oh, look at that big honking white spot.
Richard, can I just comment that the pathologist is suggesting that the radiologist do this, and why not? How would you feel about sitting there with things flashing at you, trying to make it some sort of a complicated differential diagnosis, having now for the last hour and a half worn this little hat that's pressing into your head? How does that grab you?
Depends on my Valium dose.
Yeah.
But his pigeons will do it.
Right? But you really can pick it up in overall structure in the image, right? Au contraire. I guess I'm thinking of like the like those bunch of those slides where you have like a bunch of individual cells and you're trying to
smears.
Yeah, pap smear kind of images.
Yeah, that's cytology, not pathology. Let's not get confused. With pathology, you have lots of meaningful area and lots of meaningful uninteresting area. And so one of the big challenges is to not spend time on the fat or the muscle and focus in on where the action is. So I think it's very similar to the satellite thing where you have lots of trees and ocean and stuff that doesn't matter but still fills up the data.
can I bring up a slightly different topic? So we've seen a lot of examples of different observers, and this is something that comes up a lot in medical image perception research, particularly in the area of funding, is who are your subjects going to be? Because it's difficult to get radiologists, it can be difficult and expensive. It's also difficult to get through a study section with non-trained observers, and this is an issue of where you're going to go in that balance. And so, using an example of Alejandro's data, right? So where you have all these subjects, I wonder if there isn't a happy medium where you don't just take, to just take that group of people and use that as preliminary data in a grant proposal would likely encounter difficulty in study sections, would be my suspicion. I'm not saying you're going to do that. I'm just saying if you were to. But if you went and then said, okay, we did this, we selected out the 10% of highest performers, and then we reran them again with a different kind of feedback, and we reran them again after that, and see what happens to that group, because radiologists, pathologists, people who do this in the medical context are highly selected. I mean, it's not easy to get to that position, so it's really not the same as signing up for a subject pool at a university or something like that. On the other hand, getting access to those people in sufficient numbers to prove a hypothesis is extremely difficult as well. And so somewhere, some sort of a medium has to be, a happy medium has to be found. And it would be great if there was some guidance for that, because I can tell you, as one of the people who writes those grants, sometimes you feel like it's a moving target. If you say, okay, I'm going to go to the radiologist, they say, well, that's too expensive or something. And if you say, I'm not going to go to the radiologist, they say, well, your subjects aren't well trained enough. It's a little hard to know in advance where's the sweet spot for this kind of thing, because I'm willing to take directives from outside on that. If the community says this is reasonable, then I'll work with that. If the community, and if this is unreasonable, I will avoid working with that kind of thing. But I think that issue of what your subjects are going to be is a very important one, and I hope it's something that would be useful to address at a sort of a broader level.
Let me and my pigeons jump in and really ask you guys. I mean, I think that's, I'm not in this field, but it sounds to me like they would be a good intermediate cost-reducing hypothesis testing step that might also have appeal to study sections because it's so wacky. Oh, come on.
I've got friends in the pigeon trade who swear they can't get anywhere near study sections, that they simply can't get funding for animal behavioral research, period, at this point.
Well, are they looking at clinical data?
Admittedly, they do not have their pigeons reading clinical images.
I think this may be a Rubicon that's been crossed.
Flown across.
Flown across. That's right. And
but,
you know, at low cost,
let us know how it works. And because I do have friends who would be very happy to know that somebody will fund their pigeons.
Well, I have to say we tried talking to the FDA, but they're not in an external granting situation ever. But I thought maybe as internal collaborators, but apparently we weren't doing things the right way. Yeah. Yeah.
As to using radiologists versus non-radiologist subject, I agree with what Craig is saying. There was another additional interesting wrinkle that our IRB pointed out that I think IRBs nationwide would benefit from NCI guidance, and that is, they said, if you take a trained radiologist and provide him or her feedback during training, aren't you messing with their actual clinical training? However, well, our radiologists are very protective of their training and their credentials. And so what if, somehow your training is really as effective as you think it is? What if you mess up their actual training? And so we didn't want to risk that. But then I bet, as usually happens, IRBs are kind of all over the place across institutions about issues like this. So, but is it possible for NCI to provide, or NIH, or DHHS, to provide some kind of guidance on this?
I don't know. I'm still kind of new at this. I don't really think we have much authority to tell IRBs to chill out. I wish we did. I mean, if this turned out to be a systemic problem, it could probably be addressed at the DHHS level, but I can't help you.
So.
From everything we've done with radiologists and emergency physicians, we've never had that problem with IRBs. They've never given us that complaint. I think it's also very department chair dependent. So we have chairs that say do whatever the heck you want with residents. So I think there's lots of culture.
But what Greg brings up is important because it is very recurring that we get always the same thing, and we the same thing, the one of radiologists versus trained observers. Is this that? I mean, it's the same thing. It knocks the score down, and and if it's always the same reply. We show, okay, well, there's like many papers that have radiologists and trained observers, and it's true the trained observers cannot do the clinical thing, but for the sort of stuff that we're studying, you can actually show, and there's papers showing that you can get for these simpler tasks with embedded tumors in real backgrounds, the performance can be comparable, and most importantly, if you do something to the images that improves performance of these trained observers, it will have the same effect on radiologists, except the fact that if you actually had to develop the whole thing with a radiologist, it would not ever get done because they just don't have the time, and the grant, you would be spending $200,000 in reading time for radiologists. So you can't, so it's a little bit of, but like you said, if you go with a radiologist grant, then they say it's too expensive and it's not going to be done. If you go with the thing, they say, oh, well, you don't have enough radiologists. So it's a little bit. So, and how do you solve this? Well, I think it goes back to, I think the only time I saw this try to be mitigated in some way is, like I said, there was a study, there was a 10 years ago, something like that. There's the MITB had a workshop of this type, but it actually included reviewers, because in these study sections, at least what funds this sort of stuff, there are people from physics, MDs, there are all sorts of things who have no idea about sort of perception issues and stuff like that. So they brought them on in, and they had people give a whole set of talks to basically tell them, orient them about what are the issues and what are the parameters of where these experiments or these studies exist, and that sort of helped for a few years. But now it's it's a brand new set of people, and they're raising the same issues that 10 years ago.
So I want to shift the discussion a little bit, and I want to talk about training and feedback. So yesterday when we're talking, Ragini mentioned feedback, and she said, but people said, well, you don't really know the truth in some cases. They look like masses, but you need further investigation to determine whether they are actually cancer. And then she said, well, they are a truth. But I think in these statistical learning cases, you can synthesize these backgrounds, you can synthesize the masses, and since you know that the classes from which these stimuli were created, there is actual real ground truth there. And so the question is, would radiologists benefit from such a training? Would that be something that would be worth looking at? Because this is
they benefit, and they worry that they benefit too much in the sense that my training experiments would be too effective, and it could go against their ACR training and so forth. But I do think, and we have done this in radiology residents, and it's effective, and it's just as effective as a person off the street.
They go through their regular radiology training, then they do yours, or they do yours first, and then they're listening and look at the differential scores between the radiology, standard radiology, and then
and remember that this was mentioned multiple times yesterday. Radiologists, especially radiology trainees, resident fellows, etc., are subject to assessments, and it affects their careers. So this is actually a real issue.
I think rather than talking to NIH, I think you're better off talking to the ACR. So, because I mean, ACR is very interested in like clinical decision support systems. You could try and blend this in with that, and that would encourage them to push the radiology training programs to accept it.
So along those lines, people have been proposing simulations for that very reason in medical image perception in a while. And the headwind they encounter that Andrew actually got around with their virtual clinical trials, although I imagine he can probably tell you headwind stories also, is that, okay, you're simulating. How do you know that simulation doesn't lose some critical aspect of the mammogram? The burden is on you to prove to me that that is as good as the real thing. And now you have this ground truth. And so I think those are, again, that's a recurring issue, and it's one that I don't know that there is an absolute proof that you could ever construct that would say this is as good as training on mammograms, except now we have ground truth. But maybe you don't need to. I don't know. Again, it's kind of a gray area, and it's one of the many gray areas that when you try to do this research, we try to get it funded at least, that you or when you try to get it published also, you encounter potentially different opinions of that, and you have to sort of figure out how to do it.
But you raised a good point. I was just kind of making a much more limited point. It appears that very targeted feedback is effective, and if you could come up with situations where you know the ground truth and you could, provide feedback based on absolute ground truth, that might be a component in their learning.
You're talking about harmless electric shock?
So I can't speak to training because I wouldn't recommend our model for training radiologists. But I can speak a little bit about sort of validation and realism issues. So, I mean, one of the things that gets pushed with what we're doing is there's different ways you could create the original data. So we have an algorithmic approach to simulating breast. So we actually went back to anatomy books back into the 19th century, Ashley Cooper and stuff, and we developed heuristics to simulate the anatomy of the breast. There are other groups, for example, at Duke University, that are taking CT data from John Boone and are then deforming it in certain senses to simulate de novo breasts. And there's this, well, which one's more realistic, and which one's more valid and such. And it comes down to it is be realistic to a task, or it'll be validated to a task. And this gets into what's called microsimulation modeling, which is a statistical tool for simulating, for example, clinical trials, right? So typically, if you simulate clinical trials, you're looking at groups, using group statistics. So what's the mean, standard deviation? How do it's going to branch out? The microsimulation modeling actually simulates individuals, and so then you can create these sort of much more complex sort of hierarchical trials where, okay, some individuals will go for mammography, some will go for MRI, some will go for ultrasound, depending on their breast characteristics, their predisposition to breast cancer, things like that. And so what we're doing is effectively a microsimulation, but where we're creating the images, and so we're creating women and looking at the consequences of them entering screening programs. And so what you end up having to do is do what's called calibration. And so you will choose certain criteria where you can validate the experiment against some sort of human experiment. So we'll do selected 2AFC trials or RSC trials with human observers to a particular task, demonstrate that we can calibrate our system against that task at specific points, and then basically using sort of analysis of variant, ANOVA, and other things, you can then sort of look at what's the strength of your ability to simulate uncalibrated conditions and draw conclusions from those uncalibrated conditions, and what is the well, basically the accuracy, either the trueness or the precision of those calculations based upon the number of women that you're simulating and the sort of biases that you can estimate. So then you can sort of ask de novo questions from, from this simulation. But it's hard, and it becomes very domain specific. You have to have specific paths that you're going to simulate.
All right. So on that note, I think it's time for us to take a little break, and we'll come back at 11 o'clock and start talking about the big picture. So again, I'd like to thank all of the speakers, and maybe we could all give each other a round of applause here, because I think this has been really a fabulous set of talks, and even more important from my perspective, I think the quality of the discussion has been really fantastic. So now I'd like to shift to, like I said, a slightly bigger picture. So the question is, where should this sort of this field, if we can call it, this sort of endeavor go from here? What are the big opportunities you think we've identified over the last couple of days? What kind of research have people not been doing that really needs to be done? And like I said, we could also revisit some of the issues that we were talking about yesterday in terms of what NIH is doing and what kind of advice you might have for NIH in general. So let's open up the floor.
I barely opened my mouth here. But I do have a couple of points. One is that I have pals in the optical community who are doing diffuse optical tomography, which is like fMRI, but using a bunch of fibers stuck on a skull cap, and can look at very rapidly at cortical responses to blood flow, but the time and can be combined with EEG or MEG, and it's 75% equivalent, if you will, to fMRI, functional magnetic resonance imaging, which looks at brain activity. And I think it would be very interesting to use visual to extend this into the visual perception world. And my pal Joseph Culver at University of Washington has already done DOT, what am I trying to say? Retinopathy? No, but basically retinotopy. Thank you, retinotopy. So you can actually use this to map the visual field to the cortical activation. Cheaper, faster, better, perhaps, than fMRI, and I think this is a fruitful area. So,
I think that perhaps the biggest structural issue is how to make it worth it to expert radiologists to participate in this sort of work without driving the NIH budget, into the ground. It's easy enough to get a radiologist who's interested in the project to collaborate with you, but it's really difficult to get a population of, well, readers, in effect. We can't pay them enough. We can't, the old model used to be, you looked at my images for a half hour, now you're an author on my paper. But even the radiology journals don't buy that one anymore. So are there things structurally that one could do that would make it worth significant numbers of radiologists to give us even modest amounts of time as part of what they do?
So I have a colleague that I went to school with who's now runs the accreditation program at ABR, American Board of Radiology, in Chicago. And this process has something like 100 radiologists sitting in a big auditorium, each with their own computer, looking at images and studying images. And he has offered me the ability to set up a site experiment in that same building, maybe not in that big room or perhaps even in the big room. He was looking to have some research opportunities, and so I can confirm with him that I can share his email and and put it out there.
Yeah, that would be great. One of the models that has worked is testing at ABR or testing at. We're actually running an experiment at RSNA in collaboration with one of the workstation manufacturers in next month. So that is one model that give away a free iPad on the exhibit floor. Not ideal for experimental control, but it beats nothing.
In fact, the ABR has 800 workstations in Chicago. I go there fairly often, and they're all calibrated. They're all DICOM calibrated. They're pretty much well good uniformity across them and stuff like that. So they're good experimental conditions. The lighting is variable, sound deadening, all those sort of things you'd like to have for that sort of environment.
Well, it would be great if that could be in some fashion institutionalized. Yeah. Well, I should say that we also, the gorilla experiment got us disinvited to the ABR at Louisville, sadly, which we feel very bad about because we really don't think we were saying anything bad about them. But
could I throw in it? So for serving kind of my own personal interest here, what I would love to see is efforts made at translational research that goes from vision to cancer imaging. And so we just saw today from a bunch of people in various areas of vision, lots and lots of interesting ideas. And the discussions show that there's some quirks and things about how to actually do the translation. But if there's a will to do that, I think those things are mostly overcomable, and we'll learn some new things about the perceptual processes as related to at least the detection and diagnosis.
Yeah, no, that's exactly what I'm trying to encourage here. So, but I'm sort of, I guess I'm sort of looking for something like a little more specific isn't the right word, but yeah, absolutely. We want people to start doing something that's got a more translational flavor and taking all these great ideas.
So if you want to break translational down, then to me it goes into two distinct components, and one is showing that the phenomenon is operational. That was, I think, the term I used yesterday. Is that whatever it is you've done in a different context in vision is actually functioning in the medical image perception world. And then in some regards, that's the fun part. The next part is then working out the mechanics of it, the actual nuts and bolts of it. How does it work? So I think the example we had yesterday was the context-based visual prediction for surgery. So it's one thing to say, you could easily imagine a study. I was thinking about this last night. You can imagine a study where you take those surgeons and you say, okay, here's the MRI of the brain, just not with the DTI stuff. Just how far would you cut? and you'll know they'll stop somewhere, and then you give them the tracks, and you say, okay, if I give you these tracks and tell you this is what we're measuring on DTI, how far do you cut now? And if or maybe a different group of surgeons. I'm not, the details of that experiment is something different. And then show that it's modified or something like that, and say that so this is how the actual mechanics of something creeps in and modifies what might get done. In that case, how far someone might be willing to cut in an operational setting. So those I think would be two particular parts of translation.
Problem with, I guess, that is that every kind of imaging shows a different boundary of the two. And the question, you can, and we have done that, so you can combine and collate them, and you get some kind of a shadow around. So that is what they call their level of uncertainty. And then you draw the tracks, because the tracks are just a visualization of the white matter. You can, all you want to know is they don't want to cut into the white matter, they can get it from a T1 or a T2 image. And they basically depend on the radiologist who looks at a flare or a T1 CE, a contrast enhanced, which are the very sharp boundary of the enhancement, and they say cut till the boundary. It's maximum total resection, at least in the US. So the boundary is defined by the imaging, and the imaging has diffusivity, not in the diffusion sense, but it's uncertainty as to where the boundary is. So I don't know. I think most surgeons will tell you that they will open the brain and decide. So yeah, so
well, okay. I don't think that those specific details take away from the idea that there's a perceptual process that's operational, and that there's features in those images that either the radiologist or the surgeon is using to say, this is how far you should go, or this is how far I'm going to go, and that those are the components that are modifiable in imaging, and those are the things we ought to be trying to optimize to make sure that they do this process as well as possible.
Modeling of the uncertainty is very important. So if you can give them a measure of how uncertain they are versus how uncertain the image is, that might help a lot more.
Well, could I just ask, I mean, how much of this could be done in a simulation suite? You know, all the major teaching centers now have simulation suites. We have an extensive one at Penn. A lot of the surgery that is initially done with people is done in simulation. You know, you do CPR, resuscitation, things like that in simulation. If we could think about a radiology simulation suite where we have a couple of workstations where radiologists could on a regular basis be going in, it could be maybe incorporated into their training structure in some way, that they have to spend time in the simulation suite or actually do experiments in the simulation suite. You know, part of their track is they've got to do some sort of, you know, simulation like that. I'm not quite
just
sure
like the way you
it,
make Psych 101 students do experiments.
Right, because I mean, one of the ways you could do this with you know, with what Raggedy was talking about is do it in simulation. Simulate, you know, with that feedback system, the haptic feedback system, so that, you know, you're doing a surgery, but then you can sort of say, well, okay, what if I gave you this plan? What if I gave you that plan? How does that alter your decision? You know.
Jay.
That Greg was mentioning yesterday, and that is creating some kind of national or international or maybe just national database of breast structures. It is basically a generalization of what Andrew has been doing, but it gives overall population idea, probability distribution. And I think if NCI undertook something like that, would be a tremendous resource for vision research and radiologists alike. I don't know if mammogram is the choice, the initial starting point or not. So it might be test X-rays or something like that, but it is, I think, worth doing.
Greg first, and then Brandon.
Akron has database.
Or Brandon first.
Ran a big study of images, and also has the lung imaging database consortium. So there are some resources out there if you want images.
Right. No. I mean, there are a lot of data.
So the difference being, I think what Greg was looking for yesterday, if I recall correctly, is the clear breasts, right? The negative examples, right?
Well, I was talking about both, and I think that you know, if you're looking for some really big direction that might have some big future payoffs, I think that we're a little bit on the brink of a scientific cliff here with advances in machine learning. I mean, with deep learning, these things really do seem to be able to learn just about anything. And if you really spend some time to create a good, clean data set of positives for each of these categories of pathology, and then create this hard negative data set that I was talking about yesterday that differs by about 10 orders of magnitude with regard to each of the positive samples, then just to, you know, sell the idea, say that open it up to almost a competition. saying that is open to engineers and applied mathematicians and computational vision people and computer vision people, and just tell them to throw whatever you have at it. Now, build me the best possible cancer detector and for these levels that you can make, and just see what happens. My gut tells me that would be resources very well spent.
So I think that's a great idea, but I want to sort of differentiate of what our mission is a little bit. Is our mission really to study how radiologists do this, or build sort of algorithms that replace radiologists? And to be honest, I think that there are people that are in much better position and much better skills than we are to actually do that. In fact, there are millions of dollars as a whole industry on that, millions of dollars into the whole field of computer-aided detection. No representatives here, but the whole the whole field, and maybe Brandon can comment more about that. So I think it's a great idea, but maybe you know more about this. I just don't know if we're in the position to make if that's the best place to make our contribution. I think isn't it really to re-understand the perception stuff, which is what we know. I don't know. It's just a thought. I don't know.
I guarantee you, the CAD companies are not sitting there going, I wonder what this deep learning stuff is all about. They know. They have experts. They hire people for a lot of money to go in and write their code, and they pay a lot of money to acquire lots of cases, because that's where their intellectual property is going to get developed.
Well, the other thing, too, is that in any imaging, you've got cycles of renewal and obsolescence. And so pretty much anything that you're doing on 2D mammography, by the time we all figure it out on NIH funding, we're going to be way beyond TOMO in the breast imaging. So I have to agree with Miguel and with Craig. I think it's a great opportunity, but not for this group and not for NIH.
But what I
can we get Jerry in the back there?
So you're certainly describing my life and probably Miguel's and several other people's here. That I am not an applied researcher by trade. My interest is in talking to radiologists or other experts, learning cool problems, and figuring out where that gives me leverage on the problems I care about in basic science. And in my case, one of the clear examples is the question of when do you stop searching? It goes very much to the sort of thing that Preeti was talking about. If you don't know how many targets there are, or there's seemingly nothing there, you have to make a decision about when to stop. This is something that's of high interest to radiologists who have to get through this big stack of images by the end of the day, because that's what's because the metrics that are being recorded are whether they got their cases read by 5 o'clock. So they are a lovely test bed for people who are really, really, really concerned with when's the right time to stop search so that I get my cases done, but I don't get sued for missing cancer. But what I really want to know is the more the basic science side of that. So when I'm looking for the cat, I got to figure that that's a similar kind of process, so nobody sues me if I can't find the cat. And so that sort of trade back and forth is absolutely what I want to do for a living, and I think what some of us have been trying to sell to other parts of the vision community. Greg.
Well, maybe, Andrew?
I think the other way to approach this might be with like observational studies. I mean, I'm not sure how well these work, but the eye trackers, the new ones that are not calibrated the same way that you can sort of put on the desk, can you put eye trackers into clinics? I mean, could we record what people are doing on a? It isn't, okay.
But it's a really, 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 really good idea.
Oh no, but I think it is becoming easy with the Microsoft new systems that are the tech systems. You're going to be able to. I don't know. How do radiologists typically view mammograms now? What is it against? Is it on the computer screen or?
Yeah. Computer. Yeah.
I don't think
so typically they go through a.
Right.
But they're a wearable. They're wearable eye
You don't want
trackers.
wearable ones.
You don't want the wearable.
You don't want wearable ones.
You want the one that sits on the counter and
something that you can integrate into a simulation suite. I think that can be done easily, and that shouldn't be a.
Yeah, so that's maybe another place where the simulation suite, or where you have controlled experiments. Okay, you're going to be sitting at the quiet console. You know, someone else is going to be answering the phone for you for the next hour. You know, we're going to record you. Because, and for us, you know, the sort of observational experiments are becoming more powerful. You know, especially with like like big data sort of initiatives. So we're basically, our Prosper grant is to integrate all the breast health information in the hospital into one central repository. And so we've been putting things in that really have never gone there before. So, you know, for our clinical trials work, our virtual clinical trials work, we're using the observational data of our patient population to inform our simulation studies. So we're, you know, what is the population at Presbyterian Hospital, which is one of our hospitals? Well, it's largely African American. It's, you know, serving West Philadelphia. So I can tell you, okay, they have, you know, larger breasts, lower density, you know, larger surface areas, things like that, and then I can simulate breasts like that. Whereas I can then say, oh, well, what about the main hub hospital or one of our outpatient clinics? And so I can simulate different populations of women. And if we could, you know, use these sort of observational, you can gather data observationally, you know, that might solve some of the problems of having to have these, I want to use the word lightly, contrived scenarios where you're putting people into a room for an hour and reading certain films.
So at the present time, you can potentially do this sort of desk-mounted eye tracking and keep an eye on what images they're looking at and you know, sort of statistics of the eye movements. It's much less trivial if you want to know, did they fixate on this lesion? Because you, at least, I'm not a real eye tracking expert, but I've had them in the lab. The calibration doesn't hold long enough for you to get the sort of resolution that you would need to say they fixated here, but not half a degree over, and the sort of resolution that you need in if you're looking at mammo. But this is a beautiful area where the technology has been moving fast enough that these sort of things should be possible in the, you know, not too distant future. I mean, that's, you're never going to get, we eye track radiologists, but in terms of getting them to do this in clinical practice, you're never going to be able to do that until you make it sufficiently unobtrusive that it's unobtrusive.
Can we get back to Jerry's question for a minute? I'd like maybe some of the people who are not doing research that is directly related to medical imaging to sort of comment on problems they found intriguing from the last couple of days. Yeah, Alejandro, let's start with you.
The things that I find interesting for me when I think about my theory of visual search has to do with how people are going to accrue information throughout the scene, maybe at each fixation. What's the information that is being accumulated? What are the thresholds that are driving decisions at different locations? So I think that in the past, we have a lot of us in my field have always done very positive-oriented visual search theories, meaning that we have a template and we're looking for positive evidence for that in particular regions of the display. But as it becomes evident today and yesterday from studies of the recognition of the background and where people are getting from the background, maybe we need to combine positive and negative accumulators of templateness such that we can learn to reject backgrounds, because what's unusual about this is that the backgrounds are not well structured, not like a wall. I could put my displays and on top of a scene beach, and the scene beach is not going to change anything about how fast people are finding the target in my task. But the backgrounds here are not scene beaches. They're very, and I don't think we've ever actually looked at that problem, the problem of collecting evidence for the background and or qualify it as background. Or maybe I'm wrong. But that to me was very cool and interesting, and a new problem for us from our perspective of how we develop this.
Just to follow up on that point, I mean, if you do think of a search template rather than being a template of the target, if you think of it, the template as a classification boundary, then as that classifier is learning, you would expect signal enhancement. The target should get more target-like, and the background should begin to slowly disappear.
But I guess what I'm trying to suggest is that I think that's short-sighted, and I think what I'm trying to say is that maybe we should think about multiple accumulators going towards different points, different decisions, right? So you can imagine a classifier, but instead of trying to make a binary decision, just target, non-target, do something like target, background noise, stuff in between, right? Because there's obviously those three types of things going on. I mean, obviously. This is an empirical question.
I mean, relatedly, it seems like an interesting question is how we start parsing up the scene. So we know that in visual search, if you've got things that are grouped together and form little kinesis squares, your search for them is inefficient because you've lumped them together. They've been explained away by the overall shape. And so in mammograms, the same thing is going to happen. We're in various kinds of anomalies. You're going to form larger structures. And so it seems that the joint issue of parsing the scene while searching for elements in it is generally we do visual search in either entirely parsed scenes or in very low structure noise scenes. Whereas in this case, what you've got is a bunch of overlapped structures, and so this search process is jointly parsing and searching, and that seems like a wide open and important question, both in basic perception and in actual medical image analysis.
Yeah, I definitely agree that that's one of the issues. And one of the things that struck me about what we've been talking about is it's not clear to me, when we try to think of this as a search task, which is a common way of modeling it in our world, is what are we searching over? I mean, in the kinds of tasks that we do in the models, like you were talking about, Greg, it's clear what the units are and what am I applying the template to. But in hearing about Jay's work, I'm thinking it's the whole image, right? What they were learning to do was treat that image. Is this an image with an anomaly in it? Yes or no. They're not searching over a set of defined units, or it's not clear to me that we know what they're searching, and that's the parsing problem. How does it get parsed? Are they parsing it into units and searching for, this area is a tumor and this is that, or are they? I don't know. It doesn't feel like they're necessarily doing that. It feels very different than the kinds of pre-parsed.
They're probably doing some piece of both.
Right.
One of the interesting things that's been my experience in talking to experts in a whole range of different domains is that at some point everybody will tell you that the image, whatever the class of image is, comes up on the screen, and I know this is an important one, or I know this is a bad one, or something like that. And this is the sort of thing that drives that EEG signal too. And that's presumably this global texture-y kind of signal that we don't. I mean, we don't really understand, at least anywhere near as much as we have an idea about the sort of template matching kinds of target things, but it's certainly the case that experts talk to you about that. They all think they have magic powers. Yeah, and that shows, we have that in the basic literature, well, whether or not we believe it's real, in the change here before they've localized the change.
Right. Oh, that's, yeah, I don't think. I guess I'm not from a perception researcher, so I need to ask this question. What is the aim? Is the aim to replace the radiologist, to make him better, just to understand what he's doing, or just do cool perception research? I mean, we have to kind of define that, because if you're trying to translate it to the clinic, then what do you want to achieve?
So I think when Greg's fantasy comes true and the deep learning algorithm does it all, fine, we'll go on and work on other problems. But that's not going to happen for a while. And I think in the meantime, what we're about is the human in the loop piece of it. Even with good technology, there's going to be a human who's part of that, and a human's going to have a perceptual system that we need to understand. Sure, you're making errors.
Improve or accommodate. Right.
I mean, there is a lot of it about power of suggestion. I mean, if we gave two lesions, same lesion image to two radiologists. One was the chairman and one was our chief neuro chief. Excellent. Both of them are considered excellent readers. They had a 30% difference in what they identified as lesions. 30%. The problem was the boundary, right? So then we gave them something which we extracted and suggested. And there was an aha moment for both of them. Ah, yeah, we could have considered that the boundary could have gone faster. So I'm coming back to the question that maybe it's an issue of people say that tumor boundary or tumor detection is fully solved. It's not. It's the most difficult thing to do. However, it could be both sides. You use your deep learning to get the best possible classifier of tumors so that the person perceives it. That's so it doesn't have to be just one-sided. I mean, like you said that should we do that? And people said, no, that's a classification problem. We shouldn't go into medical imaging per se. I do the tumor detection part, but I think our ground truth in our case.
Well, in the absence of the ground truth problem, which I think isn't completely intractable, and that's because there are other techniques for determining where the tumor is with molecular imaging, for example, or new fluorescent dyes that will go to a brain tumor or many other tumors and actually light them up, so you can actually see them in the operating room as glow, has a nice near-infrared glow. But I mentioned this before and got a response on the display side, but I think a tremendous opportunity is for this community to work with. I'm sort of gradually figuring out what you do, which is, I think what you do is to take the images as given and work with them. But I think there's an awful lot of pre-processing or image processing that can be done, like differential hysteresis processing, to improve the saliency of the features, and you can work with them to test and validate that the new image processing actually improves things rather than makes it worse. And so that's to me, I think where, you need to partner, I think, with the other guys who are actually messing with the images before you get them, and then see how that works.
Pre-processing or workflow even to keep track of where people look, to point them in the right direction. It kind of has feelings of CAD, but also to understand it from the vision rather than just a data mining result of CAD. I want to swing it to a little bit different issue. Obviously, I'm always thinking technology evaluation. And there's a lot of search discussions here. Most studies today have a small element of location as part of the endpoint in determining the performance of a human looking at the image. Most the time it's just, is this patient disease or not disease, because it's easier to analyze, because it's not a really well
formulated endpoints to determine how successful you are at finding the disease. So there's this tension between the clinical task, which is search, and the study, the controlled lab study, which determines and helps us understand the performance of a technology by not evaluating that they got the location right. There is plenty of good work out there trying to link the two, and many people have kind of gone with an assumption that, especially in the very controlled kind of phantom work, that if you do a study that puts the signal in the same place every time, signal known exactly study design, that the comparison of imaging techniques translates to the search problem. And that's the basic assumption, that you can do your performance evaluation with a signal known location, but it translates to the search problem, which I think there's certainly not 100% convinced opinion that is exactly transferable, but that's a link that would really help the evaluation process, either to have really good methodologies for evaluating performance in a search task and making the link to the detection task where the signal is known in a known location.
So you're saying there's a statistical challenge in quantifying localization performance as compared to quantifying classification performance. Okay.
Yes. There's lots of techniques, FROC, AFROC, LROC, but especially the industry hasn't embraced something, and it's always a very uncertain path to talk about that, because ROC and binary task is a really solid methodology that people are convinced already. Sensitivity specificity is where people normally start. You can get them to ROC, that's even better because you get more information. If you can get them to FROC, I think there's a lot of people who believe that there's even more information. And what does that mean? Smaller trials. That's really what the whole industry of medical imaging is, to do smaller trials that you understand, or at least have a good hypothesis of what's going to happen at the end. You don't do it. I mean, for research, we love to do a study and not know how it's going to end up. But for industry, they like to do studies where they know where it's going to end up. And there's some definite work there.
So some of the work that's happening in what Brandon was talking about. Oh, Miguel, did you want to go first? No. Sorry. Dev Chakravarty does a lot of work with the FROC task. And when I work with industry people, there's always this confusion about, well, he's constantly updating his website. He's constantly updating the figure of merit. So it's really tough for them to get their heads around, well, we're going to make a submission to FDA that says we're going to use the latest version of what's on Dr. Chakravarty's website. But we've had success with it. So that's one issue. Then there's also this tension between what Dr. Chakravarty is doing and what Dr. Obachowski has done, where you take an organ system and you split it into different regions of interest, which works very well with something like the colon, which has the 6 anatomical regions when you combine the flexures with adjacent regions. It's more difficult when you think about something like the breast, because that's very subjective when people are talking about the quadrants and subareolar and things like that. So there's a tension between the two of them. I guess what I'm saying is there's really good room for research in this area, and I don't know where it would come from this group or from the perception people, but it is a good open topic.
Could I zoom out a little bit?
Please.
Are we supposed to? Yeah. So I think there's a good question. Is that, what are the objectives? What are possible contributions that this group could provide? I think a good starting point is to Elizabeth Krupinski. I don't know. Are we in webinar today or no? We're not. Are we? Okay, so she might be listening. So she's written herself and Hal. There's a bunch of pieces that have been written about the priorities of medical image perception. In fact, there is such a thing called the Medical Image Perception Society. People should know about, which is a little bit small, but I would like to see. Obviously part of the reason is that there are not the resources to support vision scientists doing this type of work, and if that's going to happen, which would be great, I would like to see that group be integrated with the Image Perception Society. There are people coming from different. I mean, that's science, advances in science to me come about bringing people together and having them talk, not building parallel worlds and parallel societies. So we don't want to have the medical image perception vision society. We want to really sort of integrate those two forces, and I think looking at what their mission is a good starting point, and see how this group could add on. Obviously, it's clear what we could, what you could add is sort of what we've sort of pushed through the years is bringing vision science ideas and integrating them and bring some of that creativity and novelty, those ideas, and bring them into the problem of medical image perception, and through those ideas try to advance our understanding of how doctors do this, which might lead to sort of image quality tools, which could lead to better systems and better ways to display things, so that doctors do fewer mistakes and have just find things easier. So that's that, and then the other, instead of what comes for free, which is what Jeremy pointed out, which I also get a lot of pleasure of, is that these are complex tasks that have motivated a lot of basic science, at least I guess like Jeremy in my lab, a lot of basic science experiments to advance our understanding of how vision works. That the basic vision scientists did not run across because they're looking at T's and L's. So when you're looking at the problem of radiologists looking at tumors, there's a background, things I've been, things that it led us to think about things that the brain has to deal with in that task that otherwise you would not run across, and that enriches sort of the basic science part of visual search. So, yeah.
Craig?
I would just like to address a couple of these points. One is the relationship between the basic and applied interests, and the other is this relationship between the computer-assisted detection community and what our sort of mission should be. I think Miguel is right. I mean, I was personally hoping to learn a little bit more about what the state of the art is in that neck of the woods, and I'm I agree with Miguel that it would have been helpful if these groups are a little better mingled, so that we can talk about each other, because I believe that we really do have a I believe that it is important that we collect a whole lot of these good hard negatives so that we could start training better things. And whether that's best handled by us or best handled by them, I don't know, because I don't talk with them. And the other point that I want to make, and how those hard negatives should be collected. Maybe we have a role to play with that. Maybe it's in these simulation suites where we can, properly oversee the collection of these from radiologists using our eye tracking technology. Maybe that is our role to play. But again, I don't know enough about the state of the art to really weigh in on this at that point. With regard to the connection to the applied work and the basic work, I was delighted with what I learned here. I don't do medical imaging. I've never done it before, and I was just fascinated by the possibility that it may be possible to learn these categories of some pathologies. If there's a small number, and if we could learn what these categories are, then it would be trivial to build a sort of to make them more salient, so that they could be. Once you have the categorical target template, you could just pass it. You could just pass it over the image and highlight those reasons. Adjust your priority map. That wouldn't be a hard thing to do. And whether the radiologist would believe it and find it useful, or whether they would trust it, is a whole different question. But we could build that sort of augmented system.
I'm also not entirely sure that we are the best people to be designing computer-aided diagnostic tools, but there is a place where we could help. I think at VSS 2 years ago, somebody showed that if you have a sparse amount of data and you're trying to train a machine vision system to recognize stuff based on that, if you also train that same system to reproduce the kinds of errors that people reproduce, it yields better generalization performance, simply because the large amount of training that people have done over their lifetimes yields a specific pattern of errors, and if you can train the machine to produce those errors, that kind of induces the same kinds of structure that people have learned. So Yeah, I think you need small training examples because then you can take advantage of the human vast experience. So that's a small niche where I can see human vision helping machine vision. But generally, I'm not. I mean, there are people who are very good at using machine vision systems at doing stuff much better than we are. I think.
So just for 2 notes, I would encourage some of the vision people to try to attend some of the medical conferences, either the RSNA or the SPIE or the Medical Image Perception Society meeting, which will be next year in Belgium. That might be the closest one to sort of key your interest. But if you go to the RSNA, I mean, there's 90,000 radiologists wandering around. It's the largest trade show in the world. You'll be shocked at what it is. The second thing, though, is by and large, people are not doing CAD research anymore. Not strict at algorithm development or things like that. There are a few small groups that are doing it, just like tomosynthesis system development. I'm probably the only lab that I know of that's doing strict tomosynthesis system development. These are just too expensive. Research labs can't do them anymore. The manufacturers are doing them, and they're doing a good job. And their priorities are different than ours, perhaps. And you can inform what they do by doing very focused things, like ours work on super resolution. But ultimately, there's no hope to produce anything that's close to a clinically performing CAD system today in a research lab or a clinically performing tomo system in a research today. It's a hard task. You're up against Goliath.
Jeremy? So I think the CAD example is actually an interesting one for where this sort of a community might provide some help. Absolutely true that we're not in a position to design the CAD system at this point. But the real problem, well, not the only problem, a real problem with CAD is how little benefit it provides. And that seems to be a problem with the human in the loop. The human's good, the CAD is good, the combination is not as good as it should be. There is where we have the sort of expertise that should be relevant. Now, in the lab, you can make a CAD system with any old damn properties you want, because you can make stimuli like 3Ds and then stick a CAD signal on top of it with any known kind of validity and any prevalence that you want, and try to figure out why the humans just ain't using that signal. And so we don't need a $500 million to do the sort of work that we're good at in the CAD domain.
Good. We don't have that.
The backseat driver problem, like, yellow light up ahead, or my wife saying, there are 3 parking places over there. Well, I see them. You don't have to tell me that. But she's right. There are 3 parking places over there.
Are you suggesting that what we really need is your wife?
No, I think maybe she could learn from some of your techniques of how to develop a good CAD assisted system.
Oh, okay.
Yeah.
Well, well, there is this nice general problem that Matt may have more to say about, which is the use of the overuse, underuse of technology, and this is a specific application of it. There's certainly work on it out there. Most of it.
I also want to point out that we can also study which are the best ways to provide the CAD information, because I can tell you that from an attention perspective, if people are looking for a particular template, there's like some diffuse grayish thing or tiny white dots. Things that you're telling your attention system to do is ignore hard white boxes, ignore or red or whatever information is overlaid. That doesn't fall in your attention in your attention template. That's the sort of stuff that your attention is trying to tell you. Don't look at that. That's obviously not the target. So there is a conflict there between what your you're telling your attention to do in terms of finding the target, and the way the CAD information is delivered to the attention system. So I would imagine that if we made CAD delivery information more in tune with the attention template, maybe that's one of the sources where it's so annoying to have this information. I don't know.
So Elizabeth Krupinski, depending on how many of you are sitting on your email, says, thanks for mentioning the various research endeavors of our community and MIPS have been on the whole webinar, but it's been one way no matter how many times I use the raise hand option or chat a comment. Is there some way we can get Elizabeth in on Elizabeth, if you're listening to us, you're apparently not raising your hand correctly. I'll send you my cell phone number.
So, I wanted to say,
wait, do we have?
Elizabeth, you're unmuted also.
Hello?
Elizabeth, we can hear the background noise from your office, but we can't hear you speak more loudly.
So I also wanted to say, in my opinion, I look NIH is for programmatic direction. So I kind of see it as to some extent you guys' job to say this is what's important to us, and then we'll sort of figure out how to try and make it happen. That's a little bit the way I see it. So I'm kind of curious. So as you guys look at this, what do you think this works? Oh, that means you have her phone on.
Okay. So, I mean, to answer you, Craig, yes, absolutely. It is our job to say what are the important questions that need to be answered, and here is a pot of money to go do it, or various other mechanisms. But basically, behind that, there's an exercise of going in and figuring out where are the best ways that these two fields can collaborate, because that's my mission here. So I'm not sure exactly what I can. There's some what I can say about this in terms of yeah. Yeah, Paige, could you repeat that into a microphone?
I'll do it.
Let me, what might be a rephrase is sort of, well, okay, go ahead.
Oh, I never repeat myself the same twice. But so I think, it's really essential for you guys to give us the scientific guidance that will enable us to sort of turn your ideas as they sort of coalesce with our ideas into initiatives. So this is part of the process of us beginning to build sort of our rationale for the types of initiatives and sort of statements of interest that we will hope to release within the next year or so. So it's really critical. This meeting is not just to get together and talk about science necessarily, but it really serves a purpose in terms of helping us determine the directions that we're going to go next.
So I just wanted to put in a shameless plug for the Radiology Society's Quantitative Imaging Biomarkers Alliance, because whenever people are working with medical images, if you're doing something like, it's a total nonprofit thing, and I make no money from being involved. So
if you're doing research and you're thinking about perception and you're thinking about somebody putting a size on an image or somebody looking at diffusion coefficients in an image or uptake values in an image, there is this alliance out there. There's a website, there's a wiki. There are people who are working on how to make these numbers meaningful. And when you have your reviewers looking at grants and somebody says, oh yeah, well I'm going to do this, please make sure that they're doing it in a scientifically valid way and that they're interacting with the right people.
So another possible idea here would be, I mean, it's come up many times, you know, some of the most useful things that have been out there is like the University of South Florida mammography data set. And it's ancient. I mean, it shouldn't even be used anymore. It's inappropriate. But maybe it's time to sort of get a new data set. Maybe every decade or every 5 years or whatever, there has to be a, you know, someone's got to go out and get paid to gather in data.
Yeah. All right. So at that point, people have to run out the door and catch planes. Again, I'd like to thank everybody for the discussions. This has been fabulous. I'd like to apologize to everybody on the webinar. I guess we weren't properly integrating people's comments into the general discussion. That's just the way we set it up, and we're going to have to think about how to make that better for the next time we do this. But yes, but we love you anyway. And again, thanks to all of you for taking time out of your schedules and coming here. And as Paige has said, this was not merely an extremely fun exercise, but we are going to take your opinions seriously and start integrating them into our development of initiatives, because otherwise the initiative would just be what I think we should do, which is, you know, lots more visual search. So, yeah, exactly. Yeah. So, yeah. Thank you. All right, and again, I keep saying it, but thank you. This has been fabulous, and this is everything I imagined it would be. And have safe trips home.